Hello, dear students. I welcome you all to another masterclass session on biology. Those of you who have already attended these masterclass sessions know how and what exactly we do in these sessions. But for those who have joined this session for the first time, let me tell you what are the things that we do in this session. So, in these sessions, we basically discuss and understand the concepts which are highly important and relevant for your NEET exam, which are high yielding topics. We'll be discussing those portions over here. Now, how do we do that? In these sessions, we basically discuss the questions, and it is a question driven session. So on basis of those questions, which are very important, we discuss the related concepts. Okay. Students, we will begin with our session, and we'll be discussing those questions. Also, let me inform you that first we will be discussing questions on botany, and after that Nidhi ma'am will be joining the session, and she will be discussing some very important picked up questions for zoology. So your NEET exams is very close, and you all are doing your preparations with full swing. And with this positive energy, let us begin today's session. So the first question that we will be discussing today is here on your screen. Okay, students. So in this year's paper also, questions were asked from the binomial nomenclature, and here you can see that it is a statement based question. So the question here says, mark incorrect option regarding the scientific name of mango. So बेटा यहाँ पे आपको बायोनॉमियल नॉमेनक्लेचर के बारे में अगर पता होगा तभी आप साइंटिफिक नेम फॉर मैंगो के बारे में आंसर करेक्टली पिक कर पाएंगे. So if you look at the first option, it says Mangifera represents the genus while Indica represents the species. This is absolutely true. In biological nomenclature, if I write the scientific name of mango together, it's Mangifera indica. Now, do you see how I separately underline these scientific names? Okay, whenever these scientific names they are typed, they are italicized, but once they are handwritten, they are separately underlined. So yes, they are Latinized. They are derived from Latin language, and here the scientific name Mangifera indica. It's a binomial nomenclature. The scientific name is a binomial name. And if you look at option D, it says it should be underlined, whether handwritten or printed. No, beta, यहाँ पे handwritten तो सही है, but printed is not correct. Printed होता है तो it is just italicized. So the correct answer here would be option D. Okay. Now, बेटा इस चैप्टर से आपको और क्या चीजें याद रखनी है? क्वेश्चंस आपको ICBN and ICZN पे भी आ सकता है. They can ask you the full form of these two things. So ICBN is International Code of Botanical Nomenclature, and ICZN is International Code of Zoological Nomenclature. So these are basically universal set of codes which are given so that scientific names, biological names, binomial names can be given to both plants and animals. Okay, so clear. Let us move on to the next question. Also, students, you can give your answers in the chat box. Do drop your answers. What do you think about these questions? Okay, so we'll be discussing accordingly. Next question here talks about psychus, and it wants you to find out the correct statement with respect to psychus. In this year's paper also, beta, that you had in May, question was asked regarding gymnosperms. So gymnosperms हो बेटा इस चैप्टर में, या टेरिडोफाइट हो या ब्रायोफाइट हो, एग्जाम्पल के ऊपर आपको क्वेश्चन मिल सकता है, या फिर इन सारे जो प्लांट किंगडम के आपके मेंबर्स हैं उनके फीचर्स पे आपको स्टेटमेंट बेस्ड क्वेश्चन देखने को मिल सकता है. So if you look at the first statement over here, it says pinnately leaves persist for few years in case of cycas. Now cycas बेटा एक जिम्नोस्पर्म है, and yes, जो cycas के leaves होते हैं वो कई सालों तक persist करते हैं. For few years they are there on the plant. If I talk about B statement, it says male cone and megasporophyll. Megasporophyll is the female cone, are born on different tree. Now here, this is also a correct statement. Why? Because cycas, remember, it is a dioecious plant. And what does dioecious plant means, beta? This means that the male and female reproductive structures are present on two different plants. One possess the female cone, the other plant possess the male cone. ओके, अनलाइक पाइनस, अगर पाइनस के ऊपर बेटा क्वेश्चन आएगा तो पाइनस जो है वो मोनोइशियस होता है, सेम ट्री पे मेल एंड फीमेल कोन का प्रेजेंस होता है। क्लियर? तो आपको कंपैरेटिव जो आप क्वेश्चन देखने को भी मिल सकता है साइकस एंड पाइनस के ऊपर। लेट अस लुक एट द सी ऑप्शन. Statement here says roots have fungal association in form of mycorrhiza. नाउ बेटा जिम्नोस्पर्म्स में फंगल एसोसिएशन जरूर देखने को मिलते हैं, माइक्रोराइजल एसोसिएशन देखने को मिलता है, बट दैट इज सीन इन केस ऑफ पाइनस, नॉट साइकस। तो साइकस में क्या देखा जाता है? साइकस में बेटा हम देखते हैं कोरोलॉइड रूट्स। व्हाट आर कोरोलॉइड रूट्स? Coralloid roots are basically association of nitrogen fixing bacteria like Nostoc and Abena with the roots of Cycas. So they basically help in nitrogen fixation. Mycorrhizal root जो होते हैं बेटा ये आपका phosphorus absorption इन सब में help करेगा. Clear? So correct option क्या होगा मेरा यहाँ पे? Tell me. A and B are correct. So the correct option here would be option B. Clear? Let us move on to the next question. So next question here talks about the significance of mitosis and it wants you to select the correct option. So again एक statement based question बेटा and यहाँ पे आपको mitosis के significance के जो statements है वो देखने को मिल रही है. If I talk about A statement, it says the growth of multicellular organisms is due to mitosis. Now, this statement is absolutely true. जितने भी मल्टीसेलुलर ऑर्गेनिजम्स हैं उनमें जो ग्रोथ होती है वो माइटोसिस से देखी जाती है. माइटोसिस बीइंग एन इक्वेशनल डिवीजन. व्हाई डू वी कॉल इट एन इक्वेशनल डिवीजन बेटा? क्योंकि यहाँ पे जितना क्रोमोसोम का नंबर आपका पेरेंट सेल में होता है सेम डॉटर सेल्स के अंदर देखा जाता है. क्या हैप्लॉइड सेल्स के अंदर माइटोसिस हो सकता है? एब्सोल्युटली यस बेटा. अगर पेरेंट सेल हैप्लॉइड सेल है तो यहाँ पे भी आपका माइटोसिस देखा जा सकता है एंड इट विल फॉर्म हैप्लॉइड डॉटर सेल्स. ओके, व्हाट इज रिडक्शनल डिवीजन देन? रिडक्शन डिवीजन पे भी आपको क्वेश्चन देखा जा सकता है. सिग्निफिकेंस ऑफ मियोसिस भी आपसे पूछा जा सकता है. मियोसिस इज इन रिडक्शनल डिवीजन जहां पे नंबर क्रोमोसोम नंबर आपका हाफ हो जाता है. ओके, let us look at B statement. It says it helps in cell repair. Yes, अगर कभी भी कट लग जाता है, वून लग जाता है, डैमेज हो जाता है सेल, तो यहाँ पे सेल रिपेयरिंग जो होती है वो माइटोसिस के थ्रू की जाती है. If I talk about C statement, it says it helps to restore the nucleocytoplasmic ratio of cell. Yes, beta, nucleus is to cytoplasmic ratio cells में माइटोसिस माइटोटिक डिवीजन के थ्रू ही रिस्टोर करी जाती है. If I talk about last statement, it says it leads to variation important for evolution. No, beta, ये बात तो true है that for evolution variation is important, but this is not because of mitosis. Can you tell me which of the division does this? This is done by meiosis. ओके, सो मियोसिस की वजह से आप वेरिएशन देखोगे बिकॉज वहां पे मियोसिस में प्रोफेज वन में यू सीन की रिकॉम्बिनेशन होता है, क्रॉसिंग ओवर होता है. ओके, इन दी पेकाइटिन स्टेज. क्लियर? सो करेक्ट आंसर यहाँ पे क्या हो जाएगा बेटा? ए बी एंड सी स्टेटमेंट्स आर ट्रू. सो ऑप्शन डी विल बी आर करेक्ट आंसर ओवर हियर. क्लियर? लेट अस मूव ऑन टू द नेक्स्ट क्वेश्चन. Next question here talks about S phase. So the question says, which of the given events occur during S phase, synthesis phase of the cell cycle? नो बेटा सेल साइकिल में आपको सारे फेजेस पे जो अलग बाकी फेजेस है उन पे भी क्वेश्चन देखा जा सकता है. द अदर फेजेस आर इंटरफेस की अगर हम बात करें तो G1S G2 फेज रहते हैं एंड देन यू हैव द M फेज. सो यहाँ पे क्वेश्चन आपसे स्पेसिफिकली एस फेज के इवेंट्स के बारे में पूछ रहा है. नाउ एस फेज इज वेरी इंटरेस्टिंग. यहाँ पे बिल्कुल हमें रेप्लिकेशन ऑफ डीएनए देखने को मिलता है. यहाँ पे डीएनए का कंटेंट जो है वो बेटा डबल हो जाता है. अगर 2C कंटेंट होता है तो इट बिकम्स 4C, राइट? तो यहाँ पे आपको एक एप्लीकेशन बेस्ड क्वेश्चन भी इस तरीके से आ सकता है कि सपोज द क्वेश्चन गिव्स यू कि अगर किसी सेल में G1 स्टेज पे 20 पिकोग्राम का अमाउंट ऑफ डीएनए है तो G2 फेज में कितना अमाउंट ऑफ डीएनए रहेगा? सो यू विल डबल इट, इट विल बिकम 40 पिकोग्राम. ओके, let us look at option statement B. Duplication of semi-autonomous cell organelles. Semi-autonomous cell organelle क्या होते हैं बेटा? माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट. एंड इनका ड्यूप्लीकेशन एस फेज में नहीं होता. ओके, If I talk about synthesis of tubulin protein, this is also not done in S phase. ये G2 phase में हो रहे हैं बेटा. Okay. Duplication of centrioles in cytoplasm of higher plants. Now beta centriole duplication S phase में देखा जाता है, but not in case of plants. Plant पे नहीं बेटा. किसके अंदर? Animals में आप centriole duplication देखते हो in the cytoplasm. Clear? So यहाँ पे correct answer will be only A. Okay. Now if I talk about the chromosome number, तो chromosome number क्या होता है बेटा S phase में? The DNA content doubles, but the number of chromosome in S phase, remember beta, it remains unchanged. It remains same. अगर किसी सेल में 2n का सपोज करो 4 क्रोमोसोम्स है, तो आफ्टर एस फेज भी 4 ही क्रोमोसोम रहेगा, सेम नंबर रहेगा, 2n ही रहेगा. ओके, क्लियर? Let us move on to the next question then. Okay, so the next question talks about which of the following is true about C3 plants? Okay, C3 plants कौन से प्लांट बेटा जिनमें सिर्फ कैल्विन साइकिल आप देख रहे हो? C4 प्लांट्स क्या होते हैं? जिनमें कैल्विन साइकिल तो होता ही है, बट अपार्ट फ्रॉम दैट वहां पे क्रैंस एनाटॉमी रहती है, डिफरेंट आपको कार्बोक्सिलेशन स्टेप देखने को मिलता है इन दी बंडल शीट सेल्स एंड मीजोफिल सेल्स. ओके, सो यहाँ पे स्टेटमेंट जो ऑप्शन ए है दैट सेज दे डू नॉट शो फोटो रेस्पिरेशन. तो फोटो रेस्पिरेशन बेटा एक वेस्टफुल प्रोसेस है जो कि हमें C3 प्लांट्स में देखने को मिलता है. इसीलिए C3 प्लांट्स ज्यादा यील्डिंग नहीं होते इन कंपैरिजन टू C4 प्लांट्स. व्हाई? बिकॉज C4 प्लांट्स में फोटो रेस्पिरेशन हमें नहीं दिखता. तो ये स्टेट ऑप्शन जो है वो हमारा इनकरेक्ट हो जाएगा. They are more efficient to fix carbon dioxide as compared to C4 plants. It's the other way around beta. C4 plants are more efficient in fixing carbon dioxide in comparison to C3 plants. Okay, यहाँ पे C4 plants में rubisco carboxylase activity ही show करता है in the bundle sheet cells. Okay, there is no oxygenase activity in case of C4 plants. Option C says they have PGA as first product of carbon fixation. In C3 plants बेटा Calvin cycle अगर आप देखोगे तो वहां पे 3 steps होते हैं, carboxylation, reduction and regeneration. Now जो first stable product बनता है that is a 3 carbon compound, that's why the name C3 cycle. And what is PGA? What's the full form of PGA? PGA is 3 phosphoglyceric acid. Okay. Can you tell me what is the first stable product that you see in case of C4 plants? C4 plants में बेटा 4 carbon compound जो है वो first stable product रहता है, which is OAA, oxaloacetic acid. Clear? Last option says they show crans anatomy in their leaf. Not true, beta. This crans anatomy is a very specific and peculiar features seen in case of C4 plants. You must remember the pathway which is there in C4 plants. Clear? So the correct answer here is option C. Clear? Let us move on to the next question. So next question here talks about population interaction, and every other year you see population interaction questions in your NEET exam. So population interaction में बेटा आपको commensalism है, चाहे mutation है, चाहे predation है, parasitism है, इन पे examples पे questions आ सकते हैं. Okay, या match the column type of question आ सकते हैं जैसा कि आप अभी अपने screen पे देख रहे हैं. So try to solve this question. कमेंसालिज्म बेटा, कमेंसालिज्म अगर आप देखोगे तो ये किस टाइप का इंटरेक्शन है जहां पे दो स्पीशीज आपस में इंटरेक्ट कर रहे होते हैं. वन ऑफ द स्पीशीज इज बीइंग बेनिफिटेड बट द अदर स्पीशीज इट इज नीदर हार्म्ड नॉर बेनिफिटेड. सो यहाँ पे इट्स अ प्लस जीरो टाइप ऑफ काइंड ऑफ इंटरेक्शन. ओके, सो ये चीज कहाँ देखी जाती है बेटा? एग्जाम्पल आपको क्या याद रखना है इसमें? अगर आप ऑर्किड्स को देखोगे विच आर ग्रोइंग एज एन एपीफाइट. व्हाट आर एपीफाइट्स? जो दूसरे प्लांट पे ग्रो करते हैं. एपी मतलब ऊपर, फाइटा मीन्स प्लांट. सो मैंगो ट्री के ऊपर अगर ऑर्किड ग्रो कर रहा है, सो बेटा यहाँ पे मैंगो ट्री को कोई भी हार्म नहीं हो रहा है, बट ऑर्किड के लिए क्या हो रहा है फायदा? उसको स्पेस मिल रहा है जहां पे वो ग्रो कर सके. राइट? सो बेसिकली यहाँ पे दिस you will match with 3 orchid growing as an epiphyte on a mango tree. ठीक है. Cattle irrigates with cattle, that can also be another example जहाँ पे आपको इस टाइप का इंटरेक्शन देखने को मिलता है. B is mutualism. So mutualism बेटा, we are aware that यहाँ पे आपका positive interaction है. What does this mean? That here two species which are interacting with each other, both are getting benefited from each other. So अगर आप यहाँ list 2 देखेंगे तो बेटा कौन सा आपको ऐसा example दिख रहा है जहाँ पे both the examples are getting benefited from each other is fungi and roots of higher plant. तो ये माइक्रोराइजल एसोसिएशन जो हम पाइनस के रूट्स में देखते हैं, यहाँ पे फंजाई को रहने के लिए जगह मिल रही है. ओके, फंजाई जो है फॉस्फोरस एब्जॉर्ब करके दे रहा है, ड्रॉट कंडीशन से प्लांट को प्रोटेक्ट कर रहा है. ठीक है, एंड प्लांट इज प्रोवाइडिंग स्पेस फॉर द फंजाई टू ग्रो. ओके, if we talk about competition, now competition here is a negative interaction जहाँ पे दो species आपस में compete कर रही हैं. Okay, so correct match here would be C to 1. Okay, competition between two different species of barnacles in intertidal zone. So beta resources के लिए, space के लिए, these two barnacle species they basically compete with each other. ब्रूड पैरासिटिज्म इज अ टाइप ऑफ पैरासिटिज्म. नाउ पैरासिटिज्म बेटा एक पॉजिटिव नेगेटिव इंटरेक्शन है जहां पे आपका जो पैरासाइट है वो अपने आप को बेनिफिट करवाता है एंड होस्ट जो है वो उस पर नेगेटिव इंपैक्ट होता है. पैरासिटिज्म पे आपको एक्टो पैरासिटिज्म एंड एंडो पैरासिटिज्म भी याद रखना है. ब्रूड पैरासिटिज्म इज अ स्पेशल केस जहां पे आपके बर्ड आपको एज अ पैरासाइट की तरह बिहेव करता हुआ दिखेगा, जहां पे एग्जाम्पल है कुकू कोयल विच इज लेइंग इट्स एग इन द नेस्ट ऑफ क्रो. सो व्हाट इज हैपनिंग ओवर हियर? The cuckoo is laying its egg, and the crow is basically investing their energy in incubating the eggs of the other bird, cuckoo. Okay, so the correct match here would be A3, B4, C1, and D2. Clear? Let us move on to the next question. So next question here talks about human genome project, and it wants you to find out the statement which is not true, incorrect for human genome project. So here, if you look at option A, it says repeated sequences make up a very large portion of human genome. Yes, that is absolutely true. Repetitive sequences are in very large portions in case of human genome, and most of these sequences they do not code for any protein. Okay, so less than, if you look at option B, here it says chromosome X has most number of genes. Now beta यहाँ पे chromosome X सबसे ज्यादा genes नहीं carry करता. It is the chromosome 1 which basically carries most genes, 2968 genes. And if I talk about which chromosome carries the least, the fewer number of genes, so that is on chromosome Y. It has the fewest gene, 231 genes only. Okay. Option C says the total number of genes is estimated at 30,000. Yes, this is also true. And earlier it was thought that there were lakhs of genes which are there in humans, but it was not true. Once human genome project was done, it was seen that the total number of genes is estimated at 30,000. If I talk about option C, it says less than 2% of genome codes for protein. That is absolutely correct. Okay, so this is what you need to remember in terms of human genome project. Clear? Let us move on to the next question. Also, if the questions asks what is the largest known gene in humans, so you must remember that this is dystrophin. It has nearly 2.4 million bases. Clear? So this is also you can remember. Let us move on to the next question then. Heterochromatin is okay. So beta, in case of eukaryotes, so the chromatin is basically arranged in case of in euchromatin and heterochromatin. So heterochromatins are basically transcriptionally inactive regions. They are inactive, transcriptionally inactive, and these are darkly stained, densely packed chromatin structures. So if you look at the options, the option A here says transcriptionally active. This is not correct for heterochromatin. Yes, it is a darkly stained region. Now, the other options which are given here are basically true for euchromatin. And what does euchromatin do? Euchromatin is transcriptionally active, it is loosely packed and lightly stained. Okay. Now, let us look at the last option. It says present freely in cytoplasm. Now, beta, in case of eukaryotes, the genetic material is present inside the nucleus properly. There's a proper membrane around the nucleus. So, cytoplasm mein nahi hai, beta. It is not freely present in cytoplasm. It is there inside the nucleus. Clear? And if I talk about transcription, where does transcription take place and where does translation take place in case of eukaryotes and prokaryotes? So remember, in case of prokaryotes, because there is no nucleus, तो वहां पे क्या होगा बेटा? ट्रांसक्रिप्शन एंड ट्रांसलेशन कपल्ड जैसा रहता है एंड इट इज हैपनिंग इन दी साइटोप्लाज्म ओनली. But in case of eukaryotes, replication and transcription, this basically takes place inside the nucleus, and translation takes place in cytoplasm. Okay. So via the nuclear pore, the mRNA enters the cytoplasm and there you have translation. Clear? Let us move on to the next question then. Next question says, read the following statements and choose the correct option. Statement A says, on a chromosome where genes are tightly linked. So suppose if this is a chromosome, genes are tightly linked, placed very close to each other. So it says they will show very low recombination frequency. That is true. If two genes are tightly linked to each other, the recombination is very low. Crossing over in दो आपका जीन्स का नहीं हो पाता इतना इजीली. अगर ये जीन्स थोड़ा दूर लोकेटेड हैं, इफ दे आर लोकेटेड somewhat significant आपका इनके अंदर डिस्टेंस है, then they will show recombination. There will be crossing over during meiosis stage. Okay. So option A जो statement A जो है बेटा यहाँ पे वो correct होता है. Clear? So remember, tightly linked genes always stay together most of the time. It's and there's very less recombination. So recombination frequency भी automatically बहुत कम हो जाती है. Okay. Statement B says the dihybrid cross on Drosophila. What is Drosophila? Drosophila melanogaster is fruit fly, and it was the experimental organism which was used by Morgan. Okay. Shows higher non-parental gene combination than parental type gene combination. हाँ बेटा, मॉर्गन का जो क्रॉस एनसीईआरटी में दे रखा है जिसपे आपको क्वेश्चन नीट में पूछा जा सकता है, सो यू मस्ट रिमेंबर द डेटा व्हिच इज गिवन फॉर क्रॉस एंड क्रॉस बी. नो क्रॉस ए में आपके जो क्रॉस थे ड्रोसोफिलाज में, दैट वाज डन बिटवीन वाइल्ड टाइप एंड द म्यूटेंट टाइप, जहां पे ब्राउन बॉडी रेड आई को क्रॉस किया गया था येलो बॉडी एंड व्हाइट आई ऑर्गेनिज्म के साथ इन क्रॉस ए. सो यहाँ पे अगर आप दोनों क्रॉस देखोगे बेटा, तो दोनों में पेरेंटल टाइप का परसेंटेज वाज मच मच हायर देन दी recombinant one, the non-parental type. If I talk about cross A में बेटा 98.2% parental types थे, and if I talk about recombinant, so 98.7%, and if I talk about recombinant in case of cross A, they were 1.3%. So do you see the parental types are on much higher number? Okay, and similar situation is there in cross B. If I talk about cross B, can you tell me in cross B what was the parental percentage? In cross B, the parental type was 62.8%, and the recombinant was 37.2%. Okay, so the correct answer here would be option A, which says only statement A is correct. Clear? Let us move on to the next question. So next question is, which of the following expressions can be used to calculate the RQ? RQ is respiratory question. So in this year's NEET paper also, beta, there was an application based question where a compound was given where you need to find out the RQ value. So to do these types of question, you must first remember the formula. Okay. So RQ respiratory question ka beta jo formula hai, that is volume of CO2 evolved upon volume of oxygen consumed. राइट? अब अगर आपको डायरेक्ट क्वेश्चन यहाँ देखने को मिलेंगे तो आरक्यू ऑफ डिफरेंट कंपाउंड्स आपसे पूछे जा सकते हैं, जैसे कि अगर अपन कार्बोहाइड्रेट की बात करें, सो इन केस ऑफ carbohydrates RQ is equivalent to 1. Okay. If I talk about fats, so fats के केस में बेटा it is nearly 0.7. ट्राइपाल्मिटिक एसिड का एनसीईआरटी ने एग्जांपल दे रखा है ट्राइपाल्मिटिन का. If I talk about proteins, in case of proteins it is nearly 0.9, less than 1. Okay, so this you must remember. Let us move on to the next question. Okay, so the next question talks about ploidy level of endosperm in angiosperms. So in NEET, you will see in past year papers also from this chapter, questions are being asked on the ploidy levels of different types of cells. If they can ask the ploidy of male gametophyte, female gametophyte, synergid, central cell और endosperm. Okay. So यहाँ पे अगर अपन एंडोस्पर्म की बात करें बेटा, तो सबसे पहले हमें ये जानना है एंडोस्पर्म बनता कैसे है इन एंजियोस्पर्म्स. So this is formed as a result of triple fusion. In angiosperms there is double fertilization, and in double fertilization you have triple fusion along with syngamy. सो यहाँ पे बेटा जो आपका सेंट्रल सेल होता है जिसमें दो पोलर न्यूक्लियाई होते हैं, दे फर्स्ट फ्यूज विद ईच अदर, एंड विद दीज fused polar nuclei which now becomes diploid, the other male gamete fuses and it forms a triploid structure. So if I talk about the ploidy of endosperm in angiosperms, it is triploid. Okay. And if I talk about gymnosperms, there the endosperm ploidy is haploid. Okay. And what is the ploidy for male and female gametophyte? So the gametophytes, they are always haploid. The sporophytes are diploid. Okay. So this is how you will solve it. Clear? Let us move on to the next question. Next question says the yield of sugarcane can be increased with the help of. Now sugarcane yield जो होता है बेटा, you must memorize it. It is done with the help of gibberellins. Gibberellins आपके जो होते हैं, if you spray gibberellin on sugar canes, तो आपका of course internode जो रहता है that will elongate and lot of sugars will be accumulated over here. And अगर आप production की बात करें तो by spraying gibberellin on sugar cane, the production can be increased by 20 tons per acre. Okay. So you must also remember the various examples which are there in auxin. Auxin जो होता है बेटा आपका basically apical dominance प्रमोट करता है. एंड कैन यू टेल मी कि एपिकल डोमिनेंस को काउंटर एक्ट कौन करता है? सो साइटोकाइनिन का अगर आप प्रेजेंस देख अगर रहेगा तो इन दैट सिचुएशन लेटरल ब्रांचेस ग्रो करेंगे, एपिकल डोमिनेंस देखने को नहीं मिलेगा. ठीक है. Kinetin क्या है बेटा? Kinetin is a type of cytokinin, and there is one more name which is given in NCERT, that is zeatin. Zeatin क्या है बेटा? ये भी एक cytokinin है, but it is a naturally जो आप obtain करते हो plant से वो cytokinin है. It is obtained from zea maize. Okay, corn kernel. And kinetin कहाँ से आपका obtain किया गया था? It was obtained from herring's sperm DNA. And what is ethylene? Ethylene is a gaseous hormone which basically helps in ripening of fruit. Okay. So ethylene जो है वो आपका एक gaseous hormone है. This you must remember. Which has both, which is also shows promote growth promoter की तरह भी act करता है और inhibitor की तरह भी act करता है. Clear? Let us move on to the next question. सो यहाँ पे बेटा एवरी अदर ईयर जो क्वेश्चन है वो प्लियोट्रॉपी पे पूछा जाता है या तो फिर पॉलीजेनिक आपके जीन पे पूछा जाता है. ओके, सो यहाँ पे जो क्वेश्चन है टॉक्स अबाउट द एबिलिटी ऑफ सिंगल जीन टू प्रोड्यूस मोर देन वन फिनोटाइप. मींस सिंगल जीन एक से ज्यादा फिनोटाइप को कंट्रोल कर रही है. ओके, फिनोटाइप 1, कैरेक्टर 2, कैरेक्टर 3. सो बेटा ये जो केस है इससे हम बोलते हैं प्लियोट्रॉपी. सो दिस इज एन एग्जांपल ऑफ प्लियोट्रॉपी. पॉलीजेनिक में क्या होता है बेटा? जहां पे मोर देन वन जीन आपका कंट्रोल करता है एक सिंगल ट्रेट को. क्लियर? सो ये ऑपोजिट है. दिस इज पॉलीजेनिक इनहेरिटेंस, पॉलीजेनिक ट्रेट. ओके. So, students, this was all for your botany portion. Now, Nidhi ma'am will be taking over and she will be discussing some very important questions on zoology. So, stay tuned and I hope this session for botany was productive to you. Stay tuned.
Thank you, Khushbu ma'am, for this insightful botany session. I'm sure our students must have gained a lot of help from it. Okay. Now, after botany, it's time to dive into our zoology session. Okay. So, here I am presenting the first question onto your screens. Okay. So, as you can see, this question is match the column questions. Okay, as we discussed in our last master class, आपको याद होगा कि मैंने आपसे कहा था कि जो match the column questions है और जो statement based questions है, they have seen quite a lot of weightage in this year NEET's exam. Okay, so इसीलिए let us practice this match the column question. Okay, so this one is from human health and diseases. Biopsy, you know, biopsy is related to your histopathological studies. Okay, so A would be 3. B, non-ionizing radiation. Now you should remember कि जो आपकी UV रेडिएशन होती है वो नॉन आयोनाइजिंग रेडिएशन होती है और जो आपके एक्सरेज और गामा रेज होते हैं वो कैसे होते हैं आयोनाइजिंग रेडिएशन होते हैं ओके सो योर नॉन आयोनाइजिंग रेडिएशन इज गोइंग टू बी UV रेडिएशन नाउ बायोलॉजिकल रिस्पांस मॉडिफायर्स दिस इज योर अल्फा इंटरफेरॉन्स so C is going to be fourth and D ionizing radiation as I discussed with you it is X rays and gamma rays so this one is going to be 2 तो बच्चों हमारे आंसर्स क्या बनते हैं 3 1 4 2 ओके तो यहाँ पे 3 1 4 2 जो है वो आपका ऑप्शन सी में आता है 3 1 4 2 सो योर ऑप्शन सी इज गोइंग टू बी योर करेक्ट आंसर ओके मूविंग ऑन टू द नेक्स्ट क्वेश्चन यहाँ पे आप देखिए typhoid fever can be confirmed by which diagnostic test okay so typhoid fever can be confirmed by which diagnostic test it is vidal test है ना typhoid fever के लिए हम कौन सा टेस्ट यूज करते हैं हम vidal टेस्ट यूज करते हैं moving on okay now this one is A and R question तो ये assertion and reason question है चलो assertion पढ़ते हैं the assertion goes as the role of oxygen in the regulation of respiratory rhythm is very significant in humans okay बच्चों देखो आपको पता होना चाहिए कि अगर आप रेस्पिरेटरी रिदम के रेगुलेशन की बात करोगे देन ऑक्सीजन डज नॉट प्लेज अ वेरी वाइटल रोल इन इट ओके व्हाई बिकॉज द कीमोसेंसिटिव एरिया इन योर ब्रेन दे कैन नॉट डिटेक्ट द क्वांटिटी ऑफ ऑक्सीजन ओके what do they actually detect they detect the concentration of CO2 and H plus ions okay so there is this line in NCERT which goes as the role of oxygen in the regulation of respiratory rhythm is quite insignificant okay so assertion is going to be wrong. Now let us look at reason. A chemosensitive area situated in pons region of the hindbrain is highly sensitive to O2 and hydrogen ions. देखो ये क्यों गलत होगा? Because right now I told you the chemosensitive area it can measure CO2 and H plus ions, but it cannot measure O2. Okay, so this is wrong. Plus there are two chemosensitive area. One is central chemoreceptors, which is situated adjacent to medulla oblongata, respiratory rhythm center. Okay, and आपका जो पॉन्स रीजन है इट कंटेन्स योर न्यूमोटैक्सिक सेंटर इट डज नॉट कंटेन्स योर कीमो रिसेप्टर्स ओके सो यहाँ पे जो आपका रीजन होगा रीजन इज ऑल्सो गोइंग टू बी इनकरेक्ट तो यहाँ पे आपका आंसर क्या आ जाएगा द आंसर इज गोइंग टू बी ऑप्शन डी बोथ ए एंड आर फॉल्स मूविंग ऑन स्टेटमेंट बेस्ड क्वेश्चन स्टेटमेंट ए benign tumors do not show metastasis okay metastasis आप जानते हैं metastasis is the property जिसके through the cancer cell will spread throughout your body by traveling to blood stream okay so metastasis जो होती है it is a property of malignant tumor okay it is not a property of benign tumors so this is correct now morphine is a very effective sedative and painkiller useful after surgery okay this is true morphine जो होती है morphine is a very effective painkiller and it is usually given to patients who have undergone surgery so B is also correct okay so यहाँ पे आपका आंसर क्या आ जाएगा A because your both statement A and B are correct moving on to the next question during the post industrialization period tree trunks became dark and survival of dark winged moths increased this type of natural selection is okay देखो बच्चों जो natural selection होता है there are three types of natural selection okay one is stabilizing selection स्टेबलाइजिंग सिलेक्शन की अगर हम बात करें तो स्टेबलाइजिंग सिलेक्शन में जो आपका ग्राफ होता है वो थोड़ा सा स्टीप होता है. क्यों स्टीप होता है मिडिल से? बिकॉज द मीन वैल्यू इज सिलेक्टेड. ओके, लेकिन जो आपकी एक्सट्रीम वैल्यूज होती हैं वो डिस्कार्ड होती हैं. ओके, डिस्ट्रप्टिव सिलेक्शन की अगर हम बात करें तो डिस्ट्रप्टिव सिलेक्शन में आपके दो पीक्स बनते हैं. ओके, दो पीक्स क्यों बनते हैं? बिकॉज बोथ योर एक्सट्रीम वैल्यूज आर सिलेक्टेड वेयर एज मीन वैल्यू इज discarded. Okay. Now see this question. During the post-industrialization period, tree trunks became dark and survival of dark-winged moths increased. अगर आप इसके लिए ग्राफ बनाओगे, then आपका जो mean value होगा that would be gray. Okay. Extreme values are going to be white and black or dark. Okay. Now when you actually try to create this graph, आपको ये ग्राफ ऐसा मिलेगा where one of the extreme value is going to be selected but the other extreme value as well as your mean value is going to be discarded. What type of selection this is? This is directional selection. Okay, so the correct answer to this question is going to be option C, directional selection. Moving on, an enzyme obtained from a plant source has EC number. EC is your enzyme commission number 1214. To which of the following classes does it belong? Okay. Now, as you know, जो आपकी enzymes होती हैं, उनको 6 major classes में आप divide करते हो. Okay. And you have to remember the name of the classes. तो जो आपकी class 1 होती है, class 1 आपकी क्या होती है? Class 1 is your oxidoreductases. Okay. Class 1 is your oxidoreductases. सिमिलरली आप बात करते हो तो क्लास 2 आपकी ट्रांसफरीजेस होती है, क्लास 3 आपकी हाइड्रोलेजेस होती है. ओके, क्लास 4 जो होती है वो लाइएजेस होती है, क्लास 5 आपकी आइसोमरीजेस होती है और क्लास 6 आपकी लाइगेजेस होती है. ओके, तो व्हेन यू आर नेमिंग द एंजाइम, जो पहला नंबर होता है, द फर्स्ट नंबर, इट एक्चुअली डिनोट्स द क्लास ऑफ द एंजाइम. अब अगर यहाँ पे इस एंजाइम का क्लास 1 है, देन ऑब्वियसली इट इज गोइंग टू बिलोंग टू क्लास ऑक्सीडोरिडक्टेसेस. ओके, so an enzyme commission number is if it is 1, then it will belong to class 1, which is your oxidoreductases. Okay. Moving on. The second hard sound is associated with. Okay. दो hard sound आप पढ़ते हो. The first hard sound which is called as lub and the second hard sound which is called as dub. Okay. अब अगर आप यहाँ पे lub की बात करोगे, then lub is actually associated with the closure of AV walls. Okay. जो लब होता है विच इज योर फर्स्ट हार्ड साउंड, इट इज एसोसिएटेड विद द क्लोजर ऑफ एवी वॉल्स. जो आपका डब होता है विच इज योर सेकेंड हार्ड साउंड, इट इज एसोसिएटेड विद द क्लोजर ऑफ एसएल वॉल्स और सेमी ल्यूनर वॉल्स. ओके, तो इस क्वेश्चन में आपसे सेकेंड हार्ड साउंड के बारे में पूछा गया है. द सेकेंड हार्ड साउंड इज गोइंग टू बी डब एंड द dub is associated with the closure of semilunar walls. Okay, so option D, closure of semilunar walls, is going to be correct. Okay, now next question. The average amount of CO2 removed by lungs in an adult human under normal physiological conditions is approximately. Okay, now this question comes from the chapter excretory products and their elimination. Okay, and there is this one value which you should remember, and that is the amount of CO2 removed by lungs. It is 200 ml per minute. Okay, so this value you should remember, it is going to be 200 ml per minute. Next question. Arrange proteins A, lipids B, nucleic acid C, and carbohydrates D in descending order with respect to their average fraction in a cellular mass. Okay. Biomolecules में there is this table which actually tells you the average fraction of cellular mass of various biomolecules. Okay. And to remember that, you should remember this WPNCl. Okay. W is for water. Water constitutes your 70 to 80% of cellular mass. Okay. Next come proteins. Proteins constitute 10 to 15% of cellular mass. Next is your nucleic acids, which constitute 5 to 7%. Then you have carbohydrates, which constitute your 3%. Then you have lipids, which constitute 2%. And then you have ions. Okay. So here you have protein, lipids, nucleic acids, and carbohydrates. And we have to arrange them in descending order. So obviously protein is going to come first. After protein, there will be nucleic acid. After nucleic acid, there is going to be carbohydrates. And after carbohydrates, there is going to be lipids. So A, C, D, B. Okay. Now this is option 1. As you can see in your option 1, A, C, D, B. This is going to be your correct answer. Moving on. The switch off point of breathing which can moderate the function of the respiratory rhythm center in the medulla, is situated in which part of the brain? Okay. Now here you have to understand the switch of point of breathing. Okay. The switch of point of breathing is just another name of pneumotaxic center. Okay. जो आपका pneumotaxic center होता है उसका ही दूसरा नाम होता है switch of point for breathing. And why is pneumotaxic center called as switch of point for breathing? Because it can actually reduce the duration of inspiration. Okay, pneumotaxic center का काम क्या होता है? Pneumotaxic center can reduce the duration of inspiration and in turn it can actually increase your breathing rate. Okay, so the switch off point of breathing, which is pneumotaxic center, which reduces the duration of inspiration, is situated in the pons region of the brain. Okay, so the correct answer is going to be option A, pons. Moving on. If the cardiac output of a person is about 6.3 liter and his stroke volume is 70 ml, what will be his heart rate? Okay. See here, you have to remember one formula. What is that? It is what is your cardiac output? So your cardiac output is your stroke volume into your heart rate or heartbeat. Okay. Now you have to remember this formula. So according to this question, the cardiac output of the person is 6300 ml. Okay. The stroke volume is 70 ml, and you have to find the heart rate or heartbeat. Okay. So what is going to be your heart rate or heartbeat? You will simply divide 6300 by 70 and you will get the answer as 90 beats per minute. Okay, so what is going to be your heart rate? It is going to be 90 heartbeats per minute. Okay, now moving on. Which ART involves direct injection of sperm into the ovum? ART. What is your ART? ART is your assisted reproductive technologies which help a couple to conceive a child. Now here there is one ART which involves direct injection of sperm into the ovum. Okay. Now let us look at our options. IUI. IUI is intrauterine insemination where the semen is released into the uterine tract of female. Now, gift gamete intrafallopian transfer. It is a technique where an ovum is gifted to a female who has all the conditions to carry on pregnancy but cannot produce an ovum. So ovum from a donor is gifted to that female. ZIFT, zygote intrafallopian transfer. ZIFT is the procedure where embryos which have less than 8 blastomeres are transferred into the fallopian tube. Now, ICSI, intracytoplasmic sperm injection. Okay, if you just look at the full form or if you just look at the name of ICSI, it is very easy to deduce that what is ICSI? It is intracytoplasmic sperm injection. So cytoplasm in the cytoplasm of the ovum, sperm is directly injected. Okay, so direct injection of sperm into the ovum, that is going to be your ICSI, which is intracytoplasmic sperm injection. Moving on. Outer cell mass is involved in implantation, so inner cell mass. Okay, so this is complete the analogy question. So we have to complete an analogy. Now, outer cell mass is involved in implantation. How? Because you know this is talking about your blastocyst. In blastocyst, the outer cell mass, which is also called as trophoectoderm or trophoderm, it is involved in the formation of extraembryonic membranes, chorion, and it is involved in the implantation process. Now, inner cell mass is actually involved in the formation of embryo. Okay, so if outer cell mass is related with implantation, the inner cell mass is going to be associated with formation of embryo. So the correct answer to this question is going to be option C. Moving on. Now again we have match a column question. Okay. So in column 1 you have fetal development event and in column 2 you have time period. Okay. This is very important. This is related to your organogenesis portion from the human reproduction and you have to remember the chronology in which the fetal development takes place. Okay. Now fetal heart is formed. Fetal heart is formed after 1 month. Okay, heart is actually the first organ to develop and it is formed after 1 month. Then limbs and digit develop. Limbs and digit develop after the end of second month. Now, first movement of fetus, this takes place in fifth month. Okay, and eyelids separate and eyelashes appear. This happens at the end of sixth month. Okay, so you can say end of sixth month or end of second trimester. So this is going to be 3. So we have 4, 1, 2, 3. Now we'll just look at our options. 4, 1, 2, 3. This is your option C. Okay. So option C, 4, 1, 2, 3 is going to be your correct answer. Moving on. Statement based question. Read the statements and choose the correct option. Statement A. pre-existing advantageous mutations produce new phenotypes when selected. Okay, this is absolutely correct. Okay, जो हम एंटीबायोटिक रेजिस्टेंस वाला एक्सपेरिमेंट करते हैं बैक्टीरिया में, वहां पे हम देखते हैं कि अगर आप एक बैक्टीरियल कल्चर को किसी एंटीबायोटिक से इंट्रोड्यूस करो, then there are certain bacteria which will die, but there are also certain bacteria which will survive. Now why do they survive? Why? Because they had some mutation which made them resistant to that antibiotic. So it was a pre-existing advantageous mutation which helped them to survive. Okay. So this statement A is going to be correct. Now statement B. Natural selection enables survival and greater reproduction. Okay, this is also correct. जो आपका natural selection होता है वो survival को introduce करता है, greater reproduction भी कराता है. Natural selection was basically survival of the fittest. And what was this fitness according to Darwin? This fitness was reproductive fitness. Okay, so यहाँ पे both statements A and B are going to be correct. Okay, so option C, both statement A and B are correct. Moving on to our next question. A single stranded DNA or RNA tagged with a radioactive molecule is called one, and over 95% transgenic animals are two. Okay, so this is a question of biotechnology and its application. A single stranded DNA or RNA tagged with a radioactive molecule, this one is called as probe. Okay, so probe is a single stranded DNA or RNA which is tagged with a radioactive molecule and it is used to detect diseases in humans. Okay. Now, over 95% of transgenic animals are, what are transgenic animals? Transgenic animals are also called as genetically modified animals, and they have had their DNA manipulated to express a foreign gene. And most of the transgenic animals are mice. Okay. So your first one is going to be probe and your second one is going to be mice. So probe and mice, option C is going to be your correct answer. Moving on. Again, we have match the column question. Okay. Selectable marker. What is the function of selectable marker? Okay, you have studied two selectable marker, antibiotic resistance gene and lag Z gene. What do they help you to do? They help you to identify transformed cells. Okay, so A is going to be 3. SMA1. SMA1 is actually a restriction enzyme which produces blunt ends. Now, chilled ethanol. Chilled ethanol is added to help the precipitation of DNA. Okay, so chilled ethanol is involved with DNA precipitation, and plasmids. Plasmids have autonomous replication. Okay, so the correct answer is going to be 3, 2, 1, 4. Okay, now let us look at our option 3, 2, 1, 4. Okay, as you can see here, option D 3, 2, 1, 4 is going to be your correct answer. Moving on. Rosie is a transgenic cow producing milk rich in. Okay. So Rosie was a transgenic cow and Rosie was carrying a gene which helped her to produce milk which was actually much more nutritious than other cows' milk. Okay. So Rosie is a transgenic cow producing milk rich in human protein. Okay. It was a human protein alpha lact albumin. Okay, alpha lact albumin. So you have to remember that Rosie produced milk which was rich in alpha lact albumin and it has a quantity of 2.4 grams per liter. Okay, so Rosie, if Rosie was producing 1 liter milk, that 1 liter milk was containing 2.4 grams of alpha lact albumin, which is a very essential human protein. Okay, so the correct answer to this question is going to be option C, human alpha lact albumin. Next question. PCR involves cycling through different temperature. Which step occurs around 72 degrees Celsius? Okay. Now you know PCR की अगर हम बात करें तो PCR has three steps. One is denaturation, the second one is your annealing, and the third and last step of PCR is going to be your extension. Okay. Now let us talk about the temperature. So denaturation occurs at a temperature of more than 90 degrees Celsius. Okay, it occurs at temperature of more than 90 degrees Celsius. Annealing, this occurs at 54 degrees Celsius, and extension occurs at 72 degrees Celsius. Okay, now if we are being asked which step occurs around 72 degrees Celsius, then the answer is going to be option C, extension. Moving on. Which of the following is the most widely accepted method of contraception in India at present? Okay. So yeah, this is an NCRT line, and you have to remember that this line is actually written about a method of contraception, which is your IUDs. Okay, so you have to remember that when we talk about IUDs, we say that IUDs are actually the most widely accepted method of contraception in India at present. Okay, so the correct answer to this question is going to be option D, IUD. Okay, now moving on to the next question. During micturition, the urinary bladder blank space and the urethral sphincter blank space. Okay. Micturition, as you know, micturition is also called as urination or voiding. Okay. Micturition is also called as urination or voiding, which is basically the release of urine through urethra. Now, while micturition, the urinary bladder, the detrusor muscles of urinary bladder will contract. Okay. They will contract and your urethral sphincter will relax. Okay, so while micturition, you have to remember that your urinary bladder, you have urinary bladder and you have urethra, and you have two urethral sphincters. Okay, you have an internal urinary urethral sphincter and you have an external urethral sphincter. You have to remember that while micturition, the urinary bladder is going to contract, whereas both of your urethral sphincter, which is external and internal urethral sphincter, are going to relax. Okay, so the correct answer is contracts and relax, which is going to be option A. Okay, now moving on to the next question. Okay, select the correct statements. Presence of glucose in urine is called uremia. This is wrong. Okay, the presence of glucose in urine is going to be called as glycosuria. And uremia, what is uremia? Uremia is the increased amount of urea in blood. Okay, the increased amount of urea in blood amounting to kidney failure is called as uremia. Okay, and uremia is a very, very dangerous condition which needs to be addressed immediately. Now, distal convoluted tubule DCT selectively secretes hydrogen ions, ammonia, and potassium ions into the filtrate. This is correct. Now, presence of glucose in urine indicates diabetes mellitus. Okay, this is absolutely correct. The presence of glucose and ketone bodies in urine. Okay, presence of glucose, as we discussed, is called as glycosuria, whereas the presence of ketone bodies is called as ketonuria. Presence of both the glucose and ketone bodies in urine indicate diabetes mellitus. Okay, so your option 2 and 3 are correct, whereas your option 1 is wrong. Okay, so what are the correct statements? 2 and 3. So here option B is going to be your correct answer. Moving on, this is an A and R question. Assertion reason. Assertion. Steroid hormones directly enter the target cell and bind to intracellular receptors. Okay, this is absolutely correct. Okay, if you have a cell and here if you have a nucleus, generally nucleus will have receptors for steroid hormones. Okay, so your steroid hormones, they will directly enter into the cell and they generally bind to their intracellular receptors, which are mostly nuclear receptors. Okay, now reason. Steroid hormones are lipophilic and can easily diffuse through the cell membrane. Okay, this is absolutely correct. As you know, your plasma membrane is lipophilic. Okay, your plasma membrane, it is made up of lipids and proteins. तो आपकी plasma membrane lipids और protein से बनी होती है. तो आपकी plasma membrane कैसी होती है? Plasma membrane hydrophobic होती है. Okay, now all the hormones which are hydrophilic, they cannot pass through plasma membrane, but steroid hormones, because they are lipophilic or hydrophobic, they can easily pass through the plasma membrane. Okay, so other hormones which are, you know, lipophobic or hydrophilic, they need to generate secondary messengers, but your steroid hormones, as they can directly enter into your target cell and they can bind to intracellular receptors, they do not need to generate secondary messenger. So reason, steroid hormones are lipophilic and can easily diffuse through the cell membrane. Okay, so your assertion is also correct, reason is also correct, and reason is actually the correct explanation of assertion. So both A and R are correct, and R is the correct explanation of assertion. So your option B is going to be your correct answer. Moving on, we have match the column question. Okay, so match the following hormones with their respective diseases. Okay, insulin, as we have discussed in our previous question, insulin, deficiency of insulin will lead to diabetes mellitus. Now, thyroxine. Thyroxine, disorder in thyroxine hormone will lead to goiter. Then corticoids. Corticoids deficiency will lead to Addison's disease. And growth hormone deficiency, not deficiency, but over secretion of growth hormone in adulthood will cause acromegaly. Okay, so over secretion of growth hormone before puberty, it causes gigantism, but oversecretion of growth hormone in adult stage causes acromegaly, and deficiency of growth hormone causes dwarfism. Okay, so acromegaly is actually a disease which is associated with growth hormone. So A, 2413. Okay, 2413 is going to be your correct answer. So here option D. Option D, 2413 is going to be your correct answer. Okay, moving on. Which of the following hormones is synthesized in the hypothalamus but stored and released by the posterior pituitary gland? Okay, आपको पता है your hypothalamus has certain cells which are called as neurosecretory cells. Okay, आपके hypothalamus के पास neurosecretory cells होते हैं, और ये जो neurosecretory cells होते हैं, they secrete two hormones which are actually stored and released by posterior pituitary gland. Okay, those two hormones are ADH or vasopressin. ADH, which is your anti-diuretic hormone, and the second one is oxytocin. Okay, so the two hormones are ADH and oxytocin. So here you have option C, oxytocin. Moving on, what primary event triggers the initiation of an action potential in a neuron? Okay, initiation of an action potential actually leads to depolarization of the neuron. Okay, initiation of action potential will lead to depolarization of a neuron. और depolarization of the neuron कैसे होता है, action potential generation कैसे होता है? The action potential generation actually occurs due to influx of sodium ions. Okay, if the influx of sodium ions generates a potential change which is higher than the threshold potential, then it will actually lead to generation of action potential. Okay, so the primary event leading to the release or initiation of an action potential is influx of sodium ions. Okay, now we will be moving on to the last question of this session here. Which layer of meninges is the outermost and toughest? Okay, आपको पता है meninges जो होती है, meninges are present between skull and brain. Okay, so जो आपका cranium होता है, cranium और brain के बीच में there are meninges. Now meninges, there are three meninges present. Okay, the first meninges, this one is called as durameter. Okay, durameter is the outermost and the toughest meninges. Okay, so this one is your outermost meninges. Okay, now if I talk to you about second or middle meninges, then it is your arachnoid meter. Okay, so your arachnoid meter, this one is your middle meninges. And then your third and last meninges, which is in contact with brain tissues, which is in closest contact with brain tissue, is your pia mater, which is your innermost meninges. Okay, so you have to remember this: dura mater is the outermost, arachnoid one is the middle one, and the pia mater is the innermost one. Which this you also have to remember that dura mater is a very tough meninges, it provides protection. Pia mater is very thin, and it is in closest contact with the brain tissue out of all these three meninges. Okay, so the correct answer, which layer of meninges is the outermost and toughest, is going to be your option A, durameter. Okay, so this was all for this zoology masterclass session. I hope you are doing your preparation really well. You are reading NCRT thoroughly, and you are making sure that you remember all the examples and all the concepts correctly. Okay, so this was all for today, and I wish you all the best for your NEET exams. Keep working hard and keep studying. Thank you.